Nutrition science begins with a look at energy. The brain uses glucose exclusively as an energy source, and only in periods of starvation will the brain resort to ketone bodies. Storage energy is continuously available from 3 sources. There are small amounts of glycogen in the muscle and liver. We have sizable deposits of fat, and energy is available from the cellular mass itself, protein stores. Gluconeogenesis is the process of converting non-carbohydrate sources into glucose. This is the creation of glucose from glycerol and amino acids. The body creates energy through the process of cellular oxidation. Enzymes control these reactions. Note enzymes are proteins. Coenzymes help to activate enzymes. A number of the B vitamins serve in the production of energy. Note pantothenic acid, thiamine, riboflavin, and niacin are essential in energy production. Hormones trigger enzyme action. An example is thyroxine, which influences both physical and mental growth. It stimulates liver glycogenolysis and gluconeogenesis, so it raises blood glucose. The 2 constant reactions in energy metabolism are anabolism and catabolism. Each requires energy. Our total energy expenditure includes the basal energy expenditure, the energy expended during physical activity, and the thermic effect of food. Energy utilization can be measured in several ways. The BMR measures oxygen consumed and carbon dioxide produced. It is affected by sex and age. Note, the age group with the highest BMR is 0 to 2 years of age. A measure of the BMR is the protein-bound iodine, which measures the activity of the thyroid gland. Note, when the PBI is elevated, the basal rate is elevated. The BMR is higher during growth, pregnancy, and lactation, with a fever, and with some diseases that increase cellular activity, like cancer and hypertension. Exercise increases the rate. The resting metabolic rate is more frequently measured than the basal rate. It is greater than the basal rate. The equation that predicts most closely to indirect calorimetry is the Mifflin equation. Use actual body weight in these calculations. Following changes in someone's weight is the most practical way of measuring energy balance. Direct calorimetry measures the amount of heat produced while the body is inside a confined respiration chamber. This has limited usefulness. Indirect calorimetry uses a portable machine. This is a practical way of measuring which nutrients are being burned for energy. These metabolic carts produce data that permit calculation of the respiratory quotient. The RQ depends upon the fuel mixture being metabolized. The RQ for a pure carbohydrate intake is 1. The number of carbon dioxide molecules produced is the same as the number of oxygen molecules utilized. The RQ of a mixed nutrient intake is 0.85. Note, to lower the RQ, increase the fat intake. Carbohydrates occur as mono, di, and polysaccharides. Starch is the most significant polysaccharide in human nutrition. It is made up of glucose chains, so starch only yields glucose. Sorbitol is an alcohol derived from glucose. It is absorbed more slowly by passive diffusion. Excess sorbitol may cause diarrhea. Proteins contain carbon, hydrogen, oxygen, and are 16% nitrogen. An amino acid has both an acid and a base. Note the list of essential or indispensable amino acids. A helpful acronym in remembering this list is TV till PMH. Tryptophan is a precursor for serotonin and niacin. Complete proteins contain all the essential amino acids to maintain growth. Such proteins are referred to as having a high biologic value. Patients on low-protein diets should receive mostly HBV protein. Lipids are comprised of carbon, hydrogen, and oxygen. The state of fatty acid saturation is an important chemical characteristic. Fatty acids are saturated if all available bonds of a carbon chain are filled with hydrogen, so it is saturated with hydrogen. An unsaturated fatty acid has one or more double bonds. Note, safflower oil is the most polyunsaturated fat. Canola is the most unsaturated fat. One essential fatty acid is linoleic. Note the following. A lack of linoleic acid leads to slow growth. If linoleic acid replaces carbohydrate in the diet, LDL levels decrease, HDL levels increase. If linoleic acid replaces saturated fat in the diet, the total cholesterol, including HDL, will decrease. Safflower oil is the best source of linoleic acid. The second essential fatty acid is linolenic, essential in brain development. It comes from fish oils, walnuts, flaxseed, and canola. It decreases the hepatic production of triglycerides, but has little effect on total cholesterol levels. Fatty acids are straight hydrocarbon chains, ending in a carboxyl group and a methyl group. They are classified by the number of carbons in the chain, the position of the first double bond, and the number of double bonds. Note the location of the first double bond is counted from the methyl end of the fat and is designated by the omega sign. Hydrogenation is the reduction of a polyunsaturated fat, converting it into a saturated fat. Hydrogen is added at the double bond. Trans fatty acids have a linear structure similar to that of saturated fatty acids. Note the best sources are margarines, shortenings, and frying fats. Note the most and the least sources of the following. Coconut oil is the most saturated, canola the least saturated. Olive oil is the most monounsaturated, coconut the least monounsaturated. Safflower is the most polyunsaturated, palm kernel is the least polyunsaturated. Most heart-healthy lipids have no trans fat, no partially hydrogenated oils, and are liquid plant oils. Fats depress gastric secretions, so will delay stomach emptying. Fat has less oxygen and more carbon than carbohydrate, so provides more energy. Note, fat has more carbon atoms available for oxidation. Note, alcohol does not require digestion. It is readily absorbed through the stomach and small intestine and transported unaltered in the blood. It decreases the oxidation of fatty acids and promotes triglyceride synthesis. Alcohol is an established risk factor for head and neck cancer and may increase the risk of breast cancer. Winterized oil is useful in the preparation of salad dressings. It has been treated so it won't crystallize when refrigerated. It is a clear oil. The oils usually winterized are corn, soy, and cottonseed. On the next several pages, you will find a summary of information on vitamins and minerals. The first 4 vitamins are fat-soluble. Note the toxic level of vitamin A, its main sources, and the deficiency signs. The earliest sign of a vitamin A deficiency is nyctalopia, which is reversible. Xerophthalmia occurs after the deficiency has been severe and prolonged and is not reversible. The cornea becomes dry and inflamed. Other deficiency signs include white spots and hyperkeratosis. Vitamin D has cholesterol as its precursor. A vitamin D deficiency leads to rickets, where the bones are soft. Osteomalacia is adult rickets. Note, vitamin E is a tocopherol and is one of the least toxic vitamins. It functions as an antioxidant. Note the sources, especially cottonseed oil. Vitamin K is synthesized by bacteria of the lower intestinal tract. Note the best sources. A diet deficient in vitamin K can lead to the slow clotting of blood followed by a hemorrhage. Vitamin K is affected by anticoagulants. Patients on blood thinners should avoid changing their intake of foods high in vitamin K. The remaining vitamins are water-soluble. Note thiamine is lost in cooking as the temperature and the pH of the food rises. Its major function is in the oxidation of carbohydrate. An increase in carbohydrate in the diet requires more thiamine. Note the best sources and the symptoms of deficiency. A thiamine deficiency causes muscle weakness, foot drop, and memory loss. A laboratory value that reflects thiamine deficiency is an elevated plasma pyruvate level. Riboflavin is present in many coenzymes and is required for energy release from protein. It is destroyed by ultraviolet light, which is why milk is packaged in an opaque container. Note the best sources are animal proteins. A riboflavin deficiency affects cells with rapid turnover. Note chelosis and stomatitis, and a magenta tongue, a purple-red tongue, are symptoms. Tryptophan, an essential amino acid, is a precursor of niacin. Niacin is involved in the metabolism of carbohydrate, protein, and fat. Note, pellagra, dermatitis, dementia, and diarrhea are symptoms of a niacin deficiency. The tongue is bright red. Folate is a coenzyme essential to DNA synthesis. It prevents neural tube defects. Together with vitamin B12, it regulates the formation of red blood cells. Note the best sources include lentils and beans. Fatigue and irritability are deficiency signs. Pyridoxine acts as a coenzyme in amino acid metabolism. Note, an increase in protein requires an increase in pyridoxine. Vitamin B12 is cyanocobalamin. It contains cobalt and is bound by an intrinsic factor secreted by the stomach. It is found in animal foods, and a dietary deficiency is rare. A megaloblastic, macrocytic, or pernicious anemia may occur after a gastrectomy or removal of the ileum. Note, pantothenic acid functions as coenzyme A in energy release. Ascorbic acid is the most easily destroyed vitamin and has a structure similar to glucose. Note, it aids iron absorption. Bleeding gums are a deficiency sign. Biotin is found in many foods and is synthesized by intestinal bacteria. Calcium is the most abundant mineral in the body. Note its properties and functions. Hypocalcemia leads to tetany, which is irregular muscle contractions. Phosphorus is the second most abundant mineral. It is part of the DNA molecule. Note the best sources are animal proteins. The most absorbable form of iron is ferrous iron. Serum ferritin is the best assessment parameter for evaluating iron status. Note the absorption of non-heme iron is aided by gastric juice and vitamin C. Physical signs of iron deficiency anemia include spoon-shaped nails and pale conjunctivi. Magnesium occurs abundantly in foods. Note an increased intake of protein, calcium, or vitamin D requires additional magnesium. Zinc increases taste acuity and is found primarily in animal protein. Note it enhances insulin action. Excessive zinc intake may lead to a copper deficiency. Note its deficiency signs. Copper is essential for the synthesis of hemoglobin, and it facilitates iron absorption. It circulates as part of the protein ceruloplasmin. Low serum levels of copper are associated with Wilson's disease. Selenium functions as an antioxidant. Chromium aids insulin action and is required for maintaining glucose metabolism. Note the best sources of sulfur are animal proteins. Note as the energy expenditure of the body increases, the need for thiamine, niacin, riboflavin, and pantothenic acid also increases. Next, we'll review water, electrolytes, and acid-base balance. Body water exists in two major compartments, intracellular and extracellular spaces. Note, we lose between 0.8 and 1.2 liters of insensible water daily through the skin and breathing. An electrolyte breaks up into its ions when dissolved in water. The concentration of an electrolyte solution is expressed in milliequivalents. Note the formula used to convert milligrams into milliequivalents. The same formula can be used to convert in the opposite direction, from milliequivalents to milligrams. Sodium is the main cation in extracellular fluid. Sodium chloride is 40% sodium. Sodium is reabsorbed by aldosterone and retained by steroids. Potassium is the main cation inside the cell. Note the best and the least sources of potassium. Please note the normal blood ranges for sodium, potassium, and calcium. Water moves in and out of cells by osmosis. Diffusion occurs when particles in solution move from the more to the less concentrated side. Protein exerts colloidal osmotic pressure within blood vessels. Albumin exerts a pressure that holds the water inside. If the level of albumin drops, the pressure drops. Water leaks out of the vessel into the tissues, leading to edema. Note anasarca is extreme generalized edema, widespread swelling of the skin, associated with heart, liver, renal failure, and extreme malnutrition. Note the symptoms associated with dehydration. There will be an increase in solutes like the BUN. Most lab values will be elevated with dehydration. Serum sodium is the best assessment parameter when evaluating fluid status. Hypernatremia is associated with dehydration, hyponatremia with overhydration. A patient can be dehydrated and have edema at the same time. Water and electrolytes are also involved in maintaining acid-base balance. An acid releases hydrogen ions in solution. A base accepts or takes up hydrogen in solution. A buffer is a mixture of an acid and a base that protects the body against a strong acid or a strong base. The major extracellular buffer is a combination of carbonic acid and sodium bicarbonate. On the pH scale, which measures hydrogen ion concentration, a pH less than 7 is acidic, greater than 7 is basic. Maintaining a pH close to 7.4 is crucial for many physiologic functions and biochemical reactions. The lungs and the kidneys guard the body's acid-base balance by regulating the components of this buffer system. The lungs control the body's supply of carbonic acid. It is formed from carbon dioxide and water. Changes in the rate and depth of breathing alter the amount of carbon dioxide in the body. If the lungs hypoventilate, retaining carbon dioxide and water, the level of carbonic acid will rise. If the lungs are hyperventilating, they are losing carbonic acid. The kidneys maintain the base bicarbonate half of the buffer. They regulate hydrogen ion secretion and bicarbonate reabsorption. If the kidneys retain bicarbonate, the level of base will rise. If the kidneys excrete excess bicarbonate, the level of base will drop. The purpose of the buffer system is to maintain a pH close to 7.4. The job of the lungs and the kidneys is to return the system to balance. Note, a change in one side of the buffer brings about a compensatory change in the other side. If the level of carbonic acid rises, the kidneys compensate by reabsorbing bicarbonate. If the level of carbonic acid falls, the kidneys will excrete more bicarbonate. These changes occur readily when the lungs and kidneys are healthy. If they fail to do their job, the result is acidosis or alkalosis. If the failure is related to the pulmonary system, it is called respiratory acidosis or alkalosis. If the failure is related to the renal system, it is metabolic acidosis or alkalosis. In respiratory acidosis, there is retention of carbon dioxide and a rise in the level of carbonic acid. This may be due to hypoventilation, as in the patient with emphysema. To compensate for this rise in acid due to a failure of the lungs, the kidneys increase their absorption of base. They retain bicarbonate, excreting less in the urine. In respiratory alkalosis, there is a drop in the level of carbonic acid because the lungs have lost it through hyperventilation. To compensate for this loss of acid through the lungs, the kidneys will lose base, excreting more bicarbonate in the urine. If the situation is due to a failure of the renal system, it is metabolic acidosis or alkalosis. Metabolic acidosis is mainly due to renal disease. It may occur when the kidneys excrete too much bicarbonate. To compensate for this loss of base through the kidneys, the lungs lose acid by increasing respiration. Metabolic alkalosis may be due to retention of bicarbonate by the kidneys. The lungs compensate by retaining acid through hypoventilation. A 3-step process is used to evaluate acid-base balance. Check the patient's pH level. If the pH is dropping below 7.4, they are moving towards acidosis. If the pH is rising above 7.4, they are moving towards alkalosis. Then look at blood gas levels to determine the cause of the problem. Is it the lungs or the kidneys? The PCO2 level mainly measures carbonic acid regulated by the lungs. This tells you how well the lungs are working. HCO3 mainly measures base regulated by the kidneys, telling you how well the kidneys are performing. Note the normal HCO3 and PCO2 levels. Please proceed through the 4 patient case studies and determine the diagnosis and the compensatory response. Next, we'll review nutrient needs throughout life, beginning with pregnancy. Note the weight gain guidelines based on her BMI. The target is the lower limit of the range. Note her expected weight gain and when she is considered to be at risk. You may be given a description of several pregnant women and be asked to select who is most at risk. No alcohol should be consumed during pregnancy. Progesterone helps develop the placenta. Linolenic acid is needed for development of the fetal nervous system. Note, avoid excess intake of preformed vitamin A supplements and avoid the fish items as listed. Note the normal newborn weight begins at 2,500 grams. It is the infant age group that needs the most calories, protein, and water per unit of body weight. Please note the caloric needs: 108 cals per kilo from birth to 6 months of age, 98 cals per kilo from 7 to 12 months of age. The maternal hormones involved in lactation are prolactin and oxytocin. Prolactin stimulates the production of milk. Oxytocin moves the milk through the ducts. Colostrum meets the infant's needs during the first week of life. It has more protein than mature milk, and it has antibodies. Note, breastfeeding is recommended for at least 12 months. The milk supply is adequate if the infant is growing, has frequent stools, and 6 to 8 wet diapers per day. Note the measurable outcomes of the Baby-Friendly Hospital Initiative are to increase incidence and duration of breastfeeding. Infant formula provides the same calories as human milk. Infants need about 2½ ounces of formula per pound per day. Iron-fortified formula should be given to all infants on formula. Note the treatment for hyperbilirubinemia is hydration. Fetal iron stores decrease at 3 to 4 months of age. This is when the infant needs supplemental iron either in formula or in foods. Note, solids should be added at 4 to 6 months of age when he can hold his head and neck up and the extrusion reflex diminishes. Note, from 6 to 8 months of age, they use the palmar grasp, the whole hand. From 9 to 12 months of age, the pincer grasp enables them to pick up small pieces of food. Infants should not be fed whole cow's milk during the first year of life. No fruit juice until the age of 1. Low-fat and non-fat milks are inappropriate during the first 2 years. A sunken fontanelle may be due to dehydration or protein malnutrition. Boys and girls have the same iron needs up through the age of 10, so it is at age 11 that girls need more iron than boys. Fruit juice should be limited to 4 ounces per day up to age 6. The final growth spurt occurs during adolescence. This is the time in life when most dietary deficiencies occur. Note the adult RDA for protein in females begins at age 14. Growth is evaluated using percentile charts. Note weight for length detects short-term changes in nutritional status. The stature for age category reflects long-term nutritional status and detects stunting of growth. Note the BMI percentiles for age begin at age 2. Obesity begins at the 95th percentile. Head circumference is measured until the age of 3. Until the age of 2, they are measured lying down. Look for a change in the rate of growth as a potential risk. Failure to thrive can be caused by illness, a restricted diet, or poor appetite. A secondary cause might be a lack of fiber in the diet, which can lead to chronic constipation, poor appetite, and diminished intake. Note the symptoms of lead poisoning. Note the protein needs for adults. 56 grams for males, 46 grams for females. Fluid needs for the adult are 35 milliliters per kilogram. The nutritional requirements for the older adult are essentially the same as those for the younger adult. Calorie needs may decrease, but note, protein needs remain the same at 1 gram per kilogram. Check on the intake of calcium and iron. Folate-rich foods should be encouraged. Because of atrophic gastritis, supplements of B6 and B12 may be needed. The elderly are more vulnerable to the toxicity of vitamin A. The best advice for the athlete is to drink water. During and after continuous endurance physical activity, consume fluid, carbohydrate, and sodium. Carbohydrate loading enables the athlete to store additional glycogen in the muscle. Note, at rest and during normal activities, we burn mostly fat for energy. During prolonged exercise, carbohydrate is needed to provide pyruvate for continued fat oxidation. Creatine supplements do not enhance endurance. Non-nutritive sweeteners provide minimal to no energy and must be approved by the FDA. Sugar alcohols are karyostatic and function as humectants and emulsifiers. They are more slowly absorbed than other sugars, causing diarrhea. Some of the commonly used herbs and botanicals regulated by the Dietary Supplement Health Act are listed, with suggested primary use and possible adverse effects. Note the following have potential interactions with anticoagulants: black cohosh, garlic, ginger, ginkgo, ginseng, and St. John's wort. Prior to surgery, patients should avoid these items. Next, we will review the digestive process. Please read through the procedural breakdown. There are 3 types of digestive action. Very little chemical digestion of nutrients occurs in the stomach except for the initiation of proteolysis. Protein digestion begins in the stomach. Note, gastrin stimulates gastric secretions. Cholecystokinin contracts the gallbladder and stimulates the pancreas. Secretin stimulates the pancreas. Glucagon-like peptide 1 stimulates insulin synthesis and release. Gastric emptying of a meal usually takes between 2 and 6 hours. The acidic chyme enters the duodenum and mixes with fluids and bicarbonate ions, which neutralizes the acid. The 3rd type of digestion is bacterial and takes place in the colon. Note, the colon absorbs water, salts, and the vitamins synthesized by bacterial action. The bacteria are involved in colonic salvage, which is the anaerobic process of fermentation and absorption of the end products of carbohydrate, fiber, and amino acids. Note, bacteria convert fibers and carbohydrates into short-chain fatty acids and gases. Carbohydrates are absorbed as simple sugars in the small intestine and are then carried to the liver, where they are converted to glucose or to glycogen for storage. The body gets glucose as an energy source from the diet, from the glycogen in the liver, and from products of intermediary carbohydrate metabolism. Carbohydrates, protein, and fat all yield glucose. Note, 58% of amino acids are glucogenic. They yield glucose following deamination. Note, alanine is the most glucogenic amino acid. 10% of the fat molecule is glycerol. That is the portion of the fat that can be converted to glucose. Note, fatty acids and muscle glycogen do not add to the body's supply of glucose. Once inside the cell, glucose is oxidized to produce energy as ATP, carbon dioxide, and water. Note, these are the three end products of metabolism. Note the purpose of glycolysis, which occurs in the cytoplasm of the cell, is to produce pyruvate for the Krebs cycle. Glycolysis is the breaking down of glucose with or without oxygen into pyruvate or lactate. In aerobic glycolysis, the end product is pyruvate. In anaerobic glycolysis, the end product is lactate. Glucose enters the cell with the help of insulin. It combines with phosphorus in the presence of magnesium to form glucose 6-phosphate. It then proceeds to form pyruvic acid. The glucose 6-phosphate can also lead to the synthesis of glycogen. An important function of liver glycogen is to release glucose back to the blood at a rate to maintain normal blood sugar levels. This process requires the enzyme glucose 6-phosphatase. This enzyme functions in liver and kidney cells, not in muscle or adipose cells. So free glucose can be formed from liver glycogen and transported to other tissues for oxidation. Muscle glycogen cannot contribute to the overall body blood glucose level. It is broken down to glucose only for use within the muscle cells. The glucose 6-phosphate may also proceed through the pentose shunt, which is a side channeling of glucose that does not require energy as ATP. During this process, ribose and NADPH are produced. The pyruvic acid can proceed to form lactic acid, which is utilized by muscles when energy needs exceed the supply of oxygen, so it is produced in oxygen debt, lack of oxygen. Note the Cori cycle converts it back to glucose. But most of the pyruvic acid produced in glycolysis is converted into acetyl-CoA. Note required for this conversion are thiamine, riboflavin, niacin, pantothenic acid, and magnesium. The acetyl-CoA then becomes involved in the TCA cycle, which takes place, note, within the mitochondria of the cell. It enters not only from pyruvic acid, which is a carbohydrate, but also from fatty acids and certain amino acids. Note, acetyl-CoA is the intermediate breakdown product of all three nutrients. The TCA cycle provides most of the body's energy as ATP, releasing carbon dioxide and water. Carbohydrate is necessary to keep this process going. Oxaloacetate is the main carbohydrate fuel. It is formed from pyruvic acid and from some amino acids. When oxaloacetate reacts with acetyl-CoA, citric acid is formed. This is the first step in the cycle. If there is not adequate oxaloacetate coming in from carbohydrate to maintain the cycle efficiently, the acetyl-CoA entering from fat is not handled properly and is diverted to form ketone bodies. As carbohydrate use is decreased, the oxidation of fatty acids for energy increases. This shift to fat catabolism, plus the decreased amount of available oxaloacetate, leads to ketosis. The end products of protein digestion are water-soluble amino acids. They are absorbed rapidly from the small intestine directly into the portal blood system through the capillaries of the villi. Note pyridoxine is actively involved. Note the branched chain amino acids are valine, leucine, and isoleucine. Nitrogen balance measures body equilibrium. It compares nitrogen intake to output. A healthy individual will be either in zero or positive balance. A positive balance indicates growth. You would expect to see a positive balance in an infant, a teenager, during pregnancy, during healing. Note the formula is nitrogen in minus nitrogen out, plus a correction factor of 4, which compensates for the loss of nitrogen through the skin and the stool. A healthy person consuming a lot of protein excretes the excess nitrogen and remains in zero balance. There are several methods used to determine the quality of protein in foods: the biologic value, the net protein utilization, and the protein digestibility corrected amino acid score. If a given amino acid is not utilized in tissue protein synthesis, it may be degraded or oxidized to yield energy. Note, pyridoxine is involved. The first step is to split off the nitrogenous portion. This deamination takes place in the liver. The nitrogen group is then converted into ammonia, which is toxic. Most of the ammonia is then converted into urea and excreted by the kidneys. A small amount is used in the production of purines or through transamination in the production of the non-essential amino acids. What remains after the amino group is split off is a chain of carbons called a ketoacid. This is the part of the protein that the body burns for energy. Final fat products are formed in the intestine and bound to protein, forming lipoproteins called chylomicrons. They leave the intestine, are transported through the lymph and into the blood. Most dietary fat enters the blood as a chylomicron. The major site for lipogenesis is the adipose tissue. The liver also synthesizes fat but should not store fat. To prevent fat accumulation in the liver, lipotropic factors produce lipoproteins which transfer fatty acids out of the liver. Fat leaves the liver as a lipoprotein. Normal fat metabolism occurs in the presence of adequate carbohydrate. Fatty acids are converted into acetyl coenzyme A molecules, which then enter the Krebs cycle for oxidation. During this process, 2 acetyl coenzyme A's combine and in turn produce small numbers of ketone bodies. This is normal. The liver can use small amounts of ketone bodies for energy. Note, in certain conditions such as uncontrolled diabetes or with very restrictive intakes, carbohydrate metabolism is reducing the amount available for fat oxidation. This very long process is halted at the production of acetyl-CoA, and its production is greatly increased. These molecules combine, producing large numbers of ketone bodies, acetoacetic acid, and beta-hydroxybutyric acid. The tissues are unable to oxidize the large numbers of ketones, resulting in ketosis. Most nutrients are absorbed through active transport. The sodium pump is necessary for the intestinal and renal absorption of nutrients like glucose and certain amino acids. They move into the epithelial cell of the small intestine against a concentration gradient, necessitating both a carrier and a source of energy. In simple diffusion, water and some electrolytes diffuse from the intestine into the blood and lymph. Passive or facilitative diffusion occurs when water-soluble nutrients cannot penetrate the lipid-rich membrane of the cell, so they are attached to carriers that facilitate crossing the cell membrane. No energy is required for this process. The absorption of vitamins and minerals requires assistance from many factors. Note that vitamin D requires the acidity of chyme for absorption. it is hydroxylated first in the liver, then in the kidney. Note, folate is zinc-dependent. Food intake is centrally regulated by the glucoreceptors in the hypothalamus gland, which acts as a satiety and feeding center. Serotonin is a neurotransmitter. Note, low levels increase carbohydrate appetite. Leptin suppresses appetite. Ghrelin stimulates appetite. Note, low leptin and high ghrelin promote excess food intake. The social determinants of health include conditions in the places where people live, learn, work, and play that affect a wide range of health risks and outcomes, such as economic stability, education, access to healthy foods, safe neighborhoods, and health care. A genome is the sum of the organism's genetic material. Note, nutritional genomics focuses on diet and lifestyle-related disorders resulting from the interaction between the genome and the environment. It provides a greater understanding of how to use nutrition therapy to promote health and prevent disease.